podcast, we're going to talk about the basic structure of nucleosides, which are just the sugar and a nitrogenous base. So we're not including the phosphate in this. We're going to look at the different types of nitrogenous bases as well. All right, so on the last podcast, we talked a lot about nucleotides with a T, but now we're talking about nucleosides with an S. Nucleosides include a sugar and a base. Nucleosides have two parts. Nucleotides, as we mentioned before, are the monomers that make up nucleic acids. And nucleotides have a sugar, a base, just like nucleosides, but they also have that third part, the phosphate. The way I keep nucleosides and nucleotides straight is I remember that the word 3 starts with a T, and nucleotides has a T in it as well. So your monosaccharide can either be D-ribose or it can be D2-deoxyribose. And so the difference is whether you're dealing with DNA or RNA. In DNA, you have a deoxyribose, meaning carbon 2 is missing its hydroxyl group. It doesn't have that oxygen on the second carbon. Whereas ribose does have that hydroxyl group on carbon number 2. Both of these are aldopentoses. All right, let's talk about the second part of the nucleosides. We have the monosaccharides, but we also have a heterocyclic amine, or as most people like to call it, a nitrogenous base. So it means it's a base that has a nitrogen in it. There are two types of heterocyclic amines that are used in DNA and RNA. The first is a purine. If you have a purine, it has a double ring. So adenine and guanine are examples of purine heterocyclic nitrogenous bases. Both of them have this amino group. Both are cyclic amines, but the difference is guanine also has an amide that is not found in the adenine. The way I remember that purines are adenine and guanine is I think of silver. And the symbol for silver is Ag. And so I just remember Ag or silver is pure. And those are my purines. Pyrimidines have a single ring. So the difference here, we start with the same basic 6-carbon cyclic structure, and each of these is going to have an amine in that cyclic structure. Thymine has a methyl group, whereas cytosine, instead of having that second carbonyl, it has an amino group as well. Uracil is going to be very similar to thymine, it's just missing that methyl group. Not all of these are found in both DNA and RNA. So DNA contains the bases A, adenine, guanine, thymine, and cytosine, whereas RNA contains the bases adenine, guanine, cytosine, and instead of thymine, it uses uracil. All right, so let's talk about how we form a nucleoside. Pyrimidine bases are formed by joining the nitrogen atom at position number 1 with the anomeric carbon from our monosaccharide. When you create this linkage, this is your nucleoside. And we can name nucleosides as well. So if you have a nucleoside that comes from a pyrimidine base, it's going to be named differently than from a purine base. So nucleoside from a pyrimidine base uses the suffix IDIN. So cytosine, and we're naming it for the nitrogenous base that's attached to it, that gets changed to cytidine. If we are trying to form a nucleoside with a purine base, and remember those are your double rings, your pure silver Ag, adenine, and guanine, you're going to join the nitrogen atom at the 9 position with the anomeric carbon from your sugar, from your monosaccharide. And this is what your nucleoside would look like if your monosaccharide was bonded with a purine. So to name these nucleosides, so if you start with a purine base, you're going to use the suffix OSIN. So in this case, we have adenine. We drop the ending and it becomes adenosine. And you might recognize that, and we'll talk later in the unit about adenosine triphosphate and diphosphate. You might have heard that also from biology.